Coming today. I'm really excited for this webinar. And so today we're going to talk about how our soil biology is leading the nitrogen cycle and fueling our crop, feeding our crop and fueling our soil. So before we get going, I just wanted to introduce myself a little bit. I am a PhD student at the University of Guelph with Dr. Carrie Dunfield, but I'm actually from a farm in southwestern Saskatchewan. And if you know anything about Saskatchewan and the southwest part, you know it is pretty dry. So this is a picture that I took on our family farm. It's seeding last spring, and it looks a lot different than it does here. So moving out to Ontario has been a really fun adventure for me to learn a lot more about the crops we grow here and the difference in farming practices. And it's really exciting to see the amount of coupled livestock and cash crop farms out here. So I did my undergrad in agronomy at the University of Saskatchewan and my master's in soil science. And I like to spend a lot of my days when I'm at home on the farm, crop checking with my dad or running the combine if I'm home at harvest. But right now, as a PhD student at the University of Guelph, I am really looking at nitrogen cycling and the soil microorganisms that are driving nitrogen available to winter wheat plants. So this year was my first field season. I sampled a couple trials, Laura Van Eerd's long-term cover cropping trial, trying to understand how long-term cover cropping has linked the carbon and the nitrogen cycles together and what the microorganisms are doing in those soils and showing our plants. And I am also doing a lot of work with nitrification and urease inhibitors and trying to understand what the non-target effects are of these and how, the difference or, sorry, and how nitrification inhibitors are affecting soil nitrogen mineralization and microbial interactions with winter wheat plants and our soils. So I'm just getting into it, so you guys won't be seeing any data from my thesis, but today we're going to talk about soil microorganisms. And so maybe you've seen this figure before, it's been around for a while, but there are more organisms in 1 teaspoon of healthy soil than there are people on Earth. So there's a crazy amount of microorganisms in our soils, and our soils host 1/4 of our planet's biodiversity primarily as soil microorganisms. So we have a lot of life in our soil, and they're very diverse. And these microorganisms have a really important role in both soil health and they drive key ecosystem services. So our microorganisms are really important in our soil structure. You can see in this picture those thin little fragments on those soil aggregates. Those are actually fungi hyphae. And so they act kind of like a sticky glue to keep our soil particles together and help with our soil structure. Our microorganisms are also really important in carbon sequestration. They eat the plant litter, breaking it down, becoming some of that carbon becomes what we think of as our organic matter that provides that really nice, deep, dark color in our surface soils. And some of it gets released back to the atmosphere as CO2, completing that carbon cycle and giving plants something to eat. As part of that decomposition of plant material, that organic matter, becomes very small and integrated with our soil particles, which helps with our soil structure. It also helps absorb water to keep, our soil in place and provide water throughout the growing season to our plants when we do have times of drought like we had this year. As those microorganisms are breaking down that plant litter, they're also driving nitrogen, phosphorus, sulfur, and other micronutrient cycles because when they eat that plant litter, not only is the carbon released, but so are the nutrients that are in those plants. Microorganisms, as you know, also play a really important role in plant production. So we have microorganisms that live on every surface of the plants. Not only do they live on our roots, which is where we typically think of when we're talking about soil, they also live on the shoots and stems and leaves and seeds of our plants. And this helps plants with signaling mechanisms. So this might mean that the microorganisms are releasing chemicals that tell the plant, hey, we're running out of water. We need to take more up. We need to take up nitrogen or phosphorus. Or, hey, there's a bad bacteria here, like Fusarium or Rhizoctonia, and we think it might have some negative consequences. Maybe you need to put out some plant defense mechanisms to keep them away. Um, we really don't know all of the roles that microorganisms play in plant production and how they interact with our plants. But I do know, and I have seen when we take all of the life out of our soil through like gamma radiation, um, those plants don't grow very well. So it's important. Sorry, I clicked on something. It's important to remember that soil microorganisms hold many important roles on your farm, and they are really crucial to life. And so today we're really going to focus on that nitrogen cycling piece and how microorganisms drive our nitrogen cycle. But it's important to remember that microorganisms do much, much more than just nitrogen cycling in your soils. So we have our 4R management principles about how we manage nitrogen and how we, use our resources to the best of their abilities. And when it comes to nitrogen, really, when we're talking about managing nitrogen, we're talking about managing our soil biology. And so today we're just going to go through how nitrogen flows through soil and microorganisms and where microbes drive nitrogen transformations. Then we're going to talk about the nitrogen cycle as kind of a pipeline and how we can focus our management to ensure that our pipeline is supplying nutrients to our plants and minimizing the losses to our environment. And then we're going to talk about how we use this information kind of in our fertilizer plans, just briefly. But I'm going to get into talking about C to N ratios and how microorganisms break down plant litter and release nitrogen, how this might impact, say, cover crop management. We're going to talk about using the right source of fertilizer and what that means and how important it is to prevent our losses, and a little bit about the right place and how we know what losses we're dealing with and what we can do about that. But broadly, today, I really just want to talk about soil microorganisms and give you some background knowledge and some tools to help you think about the microorganisms on your farm. And I'm going to focus a little bit less on these new management practices most of the things that I'm going to talk about are practices that you've already heard of. And I'm not going to give you fertilizer recommendations or rates. We're just going to build our background knowledge today. Also, I forgot to mention this at the beginning of the presentation, but you guys can ask questions throughout. I can't see the chat, but Anastasia can, and so if you have questions, she'll ask them for you. Or if you have questions at the end, you'll have another opportunity to ask questions. So as we get into nitrogen cycling, it's important to keep in mind that this is co-cycled with carbon. And so there's kind of 3 major types of organisms that drive, or that we think of as microorganisms in soil and drive these nitrogen and carbon and sulfur and phosphorus cycles. The first is fungi. And so fungi are multi-celled organisms that live in long filaments, right? In that first picture, we saw those, like really skinny little stick-like figures on our soil particles. And so fungi, because they're multi-celled, they can spread out across the soil. And it means that they're also really susceptible to tillage because they can be broken up physically when we move our soil. But this also means that fungi can transport nutrients from here to our plants over here. And so they play a really important role in not only moving nutrients, but especially phosphorus, which is less mobile in our soils than nitrogen, which moves in water, and making it available to our plants just by physical translocation, and even moving water from this pore over here that seem to hold water to where our plants are living. And so we have fungi that just live broadly in our soil and are important in decomposing carbon and cycling nitrogen. But we also have fungi called arbuscular mycorrhizal fungi, or AMF, that live within our plant roots. And so they actually build their cells in the plant roots, and they exchange nutrients and they take carbon from the plants. So it's a symbiotic relationship that most plants form, but not all plants. And because those fungi take carbon, it can reduce the yield if they aren't bringing enough nutrients. But this is rare, and most plants choose to make this interaction intentionally. So while there's still a lot to discover about arbuscular mycorrhizal fungi, we do know that it's in general quite a symbiotic relationship, or quite beneficial to plants. Then we have bacteria, which are single-celled organisms, and these tend to live on soil particles. Similar to bacteria is archaea, which we know a little bit less about, and they are a little bit less sophisticated than bacteria, but both have the capacity, depending on the organism, to produce enzymes that affect our nitrogen cycle. And you can broadly think about these in two different groups. Does it fix its own carbon, like a plant does, where it photosynthesizes and takes CO2 from the atmosphere and builds it into its biomass, and that's how it eats? Those are called autotrophs. Or it eats carbon. So we as humans are heterotrophs, so are our cows, where we eat carbon that has already been made into biomass, typically by plants or some other organism. So those are called heterotrophs. And most bacteria, and really all fungi, are heterotrophic, meaning they eat carbon that is already in the soil. And so this is really important when we talk about nitrogen cycling, because the microorganisms that drive the nitrogen cycle are eating carbon. So what's going on with our carbon has a big impact on what's going on with our nitrogen. And I just broadly wanted to show you guys this to help you get thinking about where microorganisms live in soil. So these, hopefully you guys can see my pointer here. These long yellow squiggly lines are fungal hyphae. These are both the blue and the little brown particles are soil particles. The green is plant litter. And these guys are bacteria. And so bacteria typically live right on the edge of soil particles because that's where water stays. And bacteria, because they're single-celled organisms, need enough water to maintain their cell pressure. So that's why plus bacteria aren't very mobile. So they kind of get stuck on these soil particles and live in these pores. And that's why our aggregates are so important, because they help hold water in the soil and give life and home to the bacteria in our soil, who are really important in nitrogen cycling and carbon cycling and turning plant litter into organic matter. This whole picture with microorganisms driving the nitrogen cycle and eating carbon make a really complicated story when we start looking at our soil test results. And so you have your microorganisms, of course, that are living in your soil, but they're really influenced by the other abiotic factors like water and temperature and the sun, because they need enough water to live, but if there's too much, then it changes what they're doing. It's kind of like if I am on the farm versus in the lab and in the city, what I'm doing on a day-to-day basis changes. My environment changes what I'm doing. And that's the same with microorganisms. You also have the food that they're eating, so the soil organic matter and the chemistry of that organic matter changes how those microorganisms behave. And to complicate the story even further, your plants have a big impact on what microorganisms are available. And your plants secrete root exudates, which are basically candy to microorganisms that select for specific microorganisms to really live abundantly near them. And you also have multiple tiers of the soil food web where you have, say, earthworms or nematodes or protozoa that eat your microorganisms, completing this whole cycle. So as this carries on and your microorganisms, as we're going to talk about in depth later, drive your nitrogen cycle, we're trying to capture this whole, big cycle that is really impacted by a lot of factors all at the same time. And we're trying to capture that in our soil tests to predict how much nitrogen is going to be available for the growing season and how much we should supply to our plants. And so this is where understanding how the nitrogen cycle is working and what kind of factors are impacting our microorganisms can help us better manage our nitrogen and make choices around those for our management strategies of right time, right place, right source, and right rate. So in your soils, you have this pool of soil organic matter, and this is where 95% of your nitrogen is stored in your soil. This pool of soil organic matter also supplies a lot of nitrogen to your plants throughout the growing season. Even when you add fertilizer, your organic matter pool can supply upwards of 70 to 80% of the nitrogen your plants take up throughout the growing season. But at times we apply a lot of nitrogen. So what is happening? Where is all of that fertilizer going if most of it is coming from that organic matter pool? And so we'll talk about, some lost pathways, but really that organic matter pool has a really important role in making nitrogen plant available. And as you guys know, we build that organic matter pool in a number of ways, but I kind of think about plant litter decomposition feeding into organic matter. And as the microorganisms decompose that plant litter, sometimes they can release nitrogen. You also have legumes like peas or soybeans or red clover or alfalfa who form a relationship with rhizobium bacteria and fix nitrogen from the atmosphere and build it into the biomass of that plant. And as that plant decomposes, it becomes part of organic matter, and that also feeds nitrogen into your plant. So this pool of organic matter is kind of like your bank for nitrogen.
I do have a question that came in.
Yeah.
Do the herbicides we apply to control reduce the population of bacteria in the soil where the herbicides are applied?
Yeah, so that's a good question. There is varying reports on this when you read the research. It depends a bit on the herbicide and the microbes in your soil. So there are chemicals in like parts of the herbicide that your microorganisms have a really hard time decomposing, which can have negative effects. But there are also parts of your herbicide that act like that act as food. So that one's a kind of a tough question because it depends. It depends. It can, but sometimes it doesn't. And there's really no strong, straight answer. When you apply fungicides, however, which actually physically act on, microbial processes and are meant to kill microbial processes, that definitely has an impact on the fungi in your soil. And there are, non-target effects, but it is also important to keep in mind that your bacterial and fungal population in your soils readily change. Everything affects them. And so they can bounce back pretty quick. But We also have herbicides that have like a long-lasting effect, and um, our microorganisms break them down over time, and that's why the lasting effect goes away. So, sort of. That wasn't an awesome answer, but did
Well,
that?
someone followed up with fungicide and you answered it, so that was perfect.
Okay, okay, perfect. We'll keep talking about the nitrogen cycle, but if you have more questions, shout them out. So from that organic matter pool, you have bacteria that further decompose that plant litter and that organic matter and mineralize nitrogen into ammonia, which is NH4. And then you have a different pool of bacteria that then turn that ammonium into nitrate. And your plants take up ammonium and nitrate, but they do not take up soil organic matter. Plants especially prefer to take up nitrogen as nitrate, that NO3. And if you notice in that NO3 bubble, it has a little negative charge on it. And so our soils are also slightly negatively charged. So that nitrate doesn't attach to our soil profile. So it is lost more easily from our soils than ammonium. Um, Ammonium is typically the form in which AMF take nitrogen up. And so coming from this pool, typically plants take it up through AMF, although they do a little bit on their own. This is also the pool in which microorganisms go and take up nitrogen to help them decompose plant litter. And we'll talk about C to N ratios shortly, but it's important to remember that this is an immobilization. And so temporarily it takes nitrogen out of the pool that your plants are trying to also take nitrogen from, but you don't lose it from your soil. Then when you add fertilizer, again, this goes directly into those pools that your plants can take nitrogen up from, but it also means that we have potential for losses out of these pools. So these red arrows show our main loss pathways. As ammonium, you can have conversion to ammonia, and you get volatilization where this just leaves and goes into the air. From nitrate, you can have denitrification, in which bacteria specifically take those oxygen molecules away slowly, especially when there's not a lot of oxygen there, because microorganisms need oxygen and that electron acceptor to live. So it takes it further down this pathway into N2O, and ideally it becomes dinitrogen, which is what 78% of our atmosphere is made of. But sometimes it stays as this N2O. And this is why we get a lot of talk about nitrification inhibitors and policy around nitrogen fertilizer and the 30% reductions by 2030. This is really targeting these N2O emissions. These are the bad greenhouse gases, and we really don't want loss like this. We also have leaching, which is primarily something that we see here in the spring with snowmelt, where you have a lot of downward water moving. But when you're losing those nitrate molecules from your soil, it sucks. Not only are you losing money, especially if you paid money to add fertilizer into, but it also has negative environmental consequences. So I want to come back to this idea that nitrogen cycling is like a pipeline with leaky valves. So you have this pool of organic nitrogen, and your microorganisms break down that soil organic matter and they control the flow of nitrogen through the pipeline. And so that ammonium and nitrate pools are what your plants and microorganisms are competing for, but you can get losses early on as volatilization. You can get leaching from that nitrate pool and those N2O emissions. And as that pipeline carries on, if your plants don't take it up, it will continue down the line and you will have losses from your soil. And so what we are really trying to do with our management practices is make sure that we're giving our plants enough nitrogen for them to live, but not so much nitrogen that we're losing it out of our soil and losing money. So I'm going to talk about determining the right rate and kind of how our organic matter pool plays into the rates of nitrogen going on. Oh, yes. Okay, so there's kind of four situations that you can have happening in your soil. You can have a high supply and a low supply of nitrogen, a high competition where you have a lot of plant and microbe uptake, or a low competition where you don't have, a lot of plant microorganism need. And so ideally, we have soils in this green box that are balanced, they have a strong supply, we aren't losing nitrogen, but your plants are growing really well and they're getting everything you need. You can also have the blue situation. This might be like right after we add fertilizer in the spring, where you have a lot of supply, but there isn't a lot of need for that nitrogen at that time. And this is where you get leaky soils or losses from your pipelines. You can also have, the inverse relationship where you have a lot of need for nitrogen, but there isn't a lot of nitrogen availability. Maybe this is later in the growing season after your plants have taken up the nitrogen. You happen to get more rain than you were expecting. Your plants are doing really well, and there isn't a lot of nitrogen there. And so the plants are competing with the microbes for that tiny little bit of nitrogen that you need. Or you can have a balanced soil where you don't have a lot of supply coming in. So there isn't leaks and everybody's getting their needs met, but maybe not in the way that we're hoping for. So again, you can have reduced plant growth. And so all of our management practices are trying to put us in that kind of green bubble. And so we'll start with talking about how do we build a stronger supply. So you have your soil organic matter pool, and we're trying to understand and help increase the rate of mineralization into a pool that our plants can take up. We have all of these, things that are adding into our organic matter. And this is where plant litter decomposition can change our mineralization rates. So I think a lot of you have heard about C to N ratios before. And so our plant litter has a C to N ratio of about 20 to 100 carbon molecules to 1 nitrogen. And we only get a net mineralization of nitrogen initially if our ratios are less than 21 to 1, because our microbial biomass has a ratio of about 7 to 1. Bacteria are a little less, 5 to 6 carbons to 1 nitrogen. Fungi a little more, around that 8 to 9 carbons to 1 nitrogen. And these microorganisms, as they break down that plant litter, have to maintain it. But we know that our crop residues vary in C to N ratio. And so, wheat straw and corn stover has a really high C to N ratio, whereas soybean has a much lower. Our manure, cattle manure is fairly high C to N ratio, whereas say chicken manure is fairly low. And this impacts how quickly we're going to see that input of nitrogen to our soils. And so I'm just going to go through a little diagram explaining this better. So when your microorganism takes up carbon and nitrogen, they also respire carbon as CO2. So in this scenario, this microorganism has a carbon use efficiency of about 30%, a little bit over. And on average, that 30 to 40, maybe max 50% carbon use efficiency is what we see from our microorganisms. So this is the carbon they build into their biomass versus the carbon that they lose as CO2 to the air. And so again, you have another microorganism that comes along, eats more carbon, takes up nitrogen, got to maintain those ratios, respires carbon, and this leaves 2 nitrogens from that plant litter that had a really low C to N ratio for our plants to take up. So we have a net mineralization of soil nitrogen when our microorganisms decompose that plant litter. When we have a high C to N ratio, though, we get a mobilization. So that microbe, again, has to maintain their C to N ratio. They respire that carbon, leaving a C to N ratio in our plant litter of 33 to 1. And so you can have another microorganism come along, but there isn't nitrogen for them to take up. And so this is where they go to those soil nitrogen pools like ammonium and or nitrate, and they take a nitrogen molecule directly from what your plant is trying to take up, and they build it into their biomass, respiring carbon. And so this is an immobilization of soil N. So when those microorganisms die, that nitrogen returns to the soil solution for plants to take up. But initially, you have that decrease of available soil N. And so this really matters when we're thinking about when we terminate our cover crops, right? And what kind of cover crop we have. This in the picture is a radish cover crop from Laura Van Eerd's long-term cover crops down in Ridgetown, excuse me. And it has a low C to N ratio. So nitrogen is going to become available initially when that plant litter gets decomposed, and then again a little bit later on in the growing season as those microorganisms die and are decomposed, and that nitrogen gets released back into your soil solution. When you have, say, wheat straw with a really high C to N ratio, your microorganisms are going to decompose what they can with the nitrogen they have available. And then if you add fertilizer, it stimulates that microbial pool to go back and finish decomposing that wheat straw. So microorganisms, when they have lots of carbon available, will increase the competition for nitrogen in your soils. But over time, we're building our organic matter and we're building the nitrogen in our microbial biomass, and it does become available. So I'm not saying that cover crops are bad. I actually think they're a really good thing. It's just important to keep in mind. This is also kind of why if you have ever crimped your cover crop and planted directly into it, and then your crop didn't look that good after, it's because your microorganisms are so strongly competing for nitrogen in those soils initially when trying to break down that recently killed cover crop. So it's important to keep in mind this, idea of C to N ratios and what's going to become available when. Coming back to adding nitrogen into your organic matter pool, sometimes you might grow a legume specifically to add nitrogen into your soil. And so it's just important to keep in mind that some legumes are better nitrogen fixers than others. Alfalfa is going to fix a lot more nitrogen than dry bean, who is quite a poor fixer of nitrogen. So you're not going to get a lot of nitrogen added to your soil from a dry bean plant compared to an alfalfa plant. When you are growing nitrogen or legumes, it's important to keep in mind that they symbiose with rhizobia bacteria. And so you want to make sure they have the opportunity to do this. So in the picture on the left, there's a picture of faba bean roots that were grown with just our soil rhizobia, and then a picture on the right where it had fresh inoculant. And you can see that the nodules are much larger and more abundant in this right-hand picture with that fresh inoculant. But that doesn't always mean that we can't produce legumes without inoculant. You do tend to get better nitrogen fixation when you add inoculant. But in this trial, there was actually enough rhizobia present in the soil, and those nodules, while small, did fix enough nitrogen that we didn't see a difference in yield or nitrogen. When you are looking at
Sorry, there's just a question on where would oats fit in the C to N ratio?
Oh, oats. Yeah, okay. Oats also have a pretty high C to N ratio. I'm thinking they're probably around that wheat straw. They might be a little bit tighter, but they are not going to have a low enough C to N ratio where you're going to see that initial net mineralization.
Thanks.
Great. just when you're looking at your legumes, you guys probably already know this, but you want to cut open your nodules to make sure that they're red. If they're not red, your rhizobia are not actually fixing nitrogen for you. Your plants can nodulate without fixing nitrogen, so just want to make sure that you take the time to cut them open when you're looking at your plants in the spring. And when you're growing legumes, it's important. Sulfur is really important. We know this not only for legumes, but also for corn. But we don't always think about how sulfur impacts our biological nitrogen fixation. And so if you're trying to improve that soil organic matter pool or just add more nitrogen into your soils, you really want to, keep in mind that sulfur is crucial. And it has many roles in your plants, especially this signaling role, which is really important in forming that symbiotic relationship with your rhizobia bacteria so they can fix nitrogen. And traditionally in Ontario, we used to have a lot of acidic rain. So we got sulfur through our rain. With cleaner air policies, we see less of that. And so sulfur might be something you want to think about on your farm. I just want to caution that sulfur is quite variable across the field. And so testing for sulfur can be a real challenge because if you happen to sample in one of these green areas that are really high in sulfur, it can skew your whole soil test result. And you might not think that you need sulfur, but really it's only this part of the field that has enough sulfur to adequately feed your plants. Sulfur is a really tough one because it is so variable, but your agronomist can definitely help you more with those recommendations. I see there's a question, but I don't know what it says.
When is the best time to sample for sulfur?
Ooh. Sulfur is also quite mobile in our soils, just like nitrogen. So if I were living in a perfect world, I would sample for both sulfur and nitrogen in the spring because then you know what your losses through the winter were. But I understand that, it takes a while to get test results back from the lab. So in an ideal world, you sample right before seeding, you know exactly what was in your soil, and then you go and seed and apply your sulfur down with your seed. But I understand that's not reasonable in a lot of cases. So sampling, typically when we sample, we do just sample, our soils for all of our nutrients at one time and try and get a snapshot. But yeah, again, sulfur is a tough one to sample for, primarily in space, but also in time.
There's another question on is organic matter and sulfur directly correlated?
Oh, that's a good question. I have not looked that deep into sulfur cycling, so I actually don't know. Yeah, I can get back to you on that, but sorry, I didn't think of that one.
Oh, and I will leave my email, like the research email, and then I can connect you with Amanda after and see if we can get the questions there. There's also another question that appeared. For soybeans following corn needing additional sulfur, when would be the best time to apply our sulfur?
Um, okay, yeah, applying with sulfur again. The big thing is you can also have sulfur losses. Um, so it's best that you get it in the ground. So while in general, the best time to apply any nutrient is exactly when your plant needs it, which would be, after your plant has germinated and it's, kind of greening up. But because you can have those losses of sulfur, just like you can have gaseous losses of nitrogen, applying it in the spring, getting it in the ground is really probably your best bet. Are we good for questions?
We're good.
Okay. Uh, Moving on, just coming back to this, building your soil organic matter. And in some cases, you have strong soil organic matter and you don't need to build, you just aim to maintain helps your nitrogen bank. So that means minimizing your soil disturbance. So your bacteria and fungi have a home to live in as much as you can on your farm. Again, I understand that we're all limited by the tools we have, and tillage is a tool that we do use. Of course, no-till is always ideal for your microorganisms, but I understand there's limitations. Keeping your plants in the ground year round and diversifying what plants you're growing means feeding your microorganisms and feeding a broader population of microorganisms growing different plants. Applying carbon inputs, whether that's manure or other organic amendments, can be really beneficial. And can you integrate livestock? Grazing has, a number of benefits. Manure especially has a lot of benefits, and sometimes if you can include livestock, that allows you, the ability to have some perennialization where you grow alfalfa for a couple of years. And so those principles are really just, part of this triangle of soil health. I'm not sure if you guys have seen this before, but the goal of when we talk about soil health, it's a balance of maximizing your carbon gains and minimizing your carbon losses. And so this came right out of a textbook that Laura Van Eerd was a part of publishing. So again, really just those principles that I talked about, really thinking about how you can take care of your organic matter pool. When we're talking about making choices around what rate to apply, we have this pool of organic matter that we know is going to supply nitrogen, but how much nitrogen it supplies throughout the season and when it supplies is really linked to your water and your temperature. When you have quite a bit of water in your soils, so your soils are not quite completely saturated, but they're above field capacity, you have a lot of mineralization to ammonium occurring. So this is that movement from soil organic matter into a more available nitrogen pool that your plant can take up. And when you decrease the water a little bit, so just above field capacity, you have nitrification occurring. And so this is directly because of the microorganisms that are able to live in these conditions and what genes are turned on based on how much water is there and how much oxygen they're able to receive. But when it's really wet, microorganisms don't have enough oxygen to oxidize nitrogen into nitrate. So this is where, we don't know when it's going to rain and we don't know how much it's going to rain. It would be amazing if we could, and it would really help us predict how much nitrogen is going to become available through the growing season. But we're all trying to manage that. And we know that it is linked to our soil organic matter. And so we do have some soil testing labs do try and estimate what's going to become available, and they'll give you fertility recommendations. And that they're really taking into account, what the seed-to-N ratio of your previous crop that you grew was, how frequently you grow crops. As you can see, this is a fertility recommendation from Saskatchewan for canola and wheat. That's all I had. Sorry, guys, I don't have data off a Ontario farm, but this is just to give you an idea of what is feeding into your soil test recommendations. And so I'm going to move on from determining your right rate to talking about after you add your fertilizer, what happens in your soil and how do you know your losses. So when we talked about how different amounts of water in your soil really impact what nitrogen is going to be most available and what microorganisms are going to be active, it comes back to this core principle that nitrogen cycling is all about electron transfers. So microorganisms are oxidizing and reducing nitrogen all of the time. And the key part of this, my main point, is really when you move from ammonium to nitrate, you have this release of hydrogen, and hydrogen is a proton that's important in pH. And I don't have a scale for pH here, but an acidic pH has a greater balance of hydrogen ions compared to OH or hydroxyl ions. And so the more hydrogen ions in your soil, the more acidic your pH is. And so this is where choosing the right source, and as you're going to see, minimizing your losses becomes really important. So when you apply ammonium-based fertilizers, these contribute to the acidification of your soil, whereas urea and nitrate and ammonia-based fertilizers don't. And I'll show you how this works. So when you apply a MAP, you go from ammonium to nitrate, and that conversion releases 2 hydrogen ions, but you have 1 molecule of ammonium and 1 molecule of nitrate. Your plant then takes up that nitrate, and it has to maintain its electrical balance, so it releases a hydroxyl ion, which is negatively charged, and this hydroxyl ion gets together with that hydrogen and forms a water. But that other hydrogen ion here is still present in your soil, and it gets released. If you happen to lose that nitrate ion, though, whether that's through denitrification or leaching, both of those protons or hydrogen ions go into your soil. When you have urea, you don't have that acidification effect because it takes a hydrogen to convert urea into ammonium. So it all balances out where, again, you get 1 molecule of water out of this, but that hydrogen ion intake balances out that hydrogen ion loss. But if you lose that nitrate molecule, again, you have both of those hydrogen ions going into your soil solution, contributing to soil acidification. And using soil organic matter nitrogen works very, very similarly to that urea, where again, it took a hydrogen ion and you had 1 release, so it's a net 0. Unless you lose that nitrate. If you lose that nitrate out of your soil, you have that acidification. So as you can see, even, choosing the right source that is going to feed your plants is of course important, but especially keeping that nitrate in your soil is really, really crucial for not having this acidification effect. And so this is based on ultimately mosaic crop nutrition output. So urea, UAN, anhydrous ammonia, because it took that hydrogen input, had no net acidification. CAN, sodium nitrate, also comes in as nitrate, so no acidification effect. It actually has a bit of a liming effect. But MAP and diammonium phosphate can lead to net acidification. Okay, so we went through the right source and how important it is that we prevent losses of our nitrate to not have acidification of our soils. And of course, when you have acidification of your soils, you know that you have to apply lime, which is really, really expensive to try and mediate that. So ideally, we never get to a point where we have, soil acidification going on. Moving on to right time, this is where we really work to close those leaky valves. So we want to really time the supply of nutrients to our plants with when they need them. And so, yes, we have these abiotic conditions like temperature and moisture that influence what's happening in our bacteria. And ideally, during times when we have a lot of freeze-thaw cycles in the spring, we don't have a lot of nitrogen sitting there because we have a lot of losses from our bacteria during this time. It is crazy what is happening. Bacteria are dying, and then, there was this flush of nutrients, so more bacteria are living, and it gets quite active. So this is one of the key reasons why we don't want to be applying nitrogen in the fall, is because in these freeze-thaw cycles, we get a lot of these cell lysing and a lot of N2O emissions. And so not only are do we, do some split application to try and help with timing nutrients to our plant needs, we can also use inhibitors. And so we have urease inhibitors that prevent this conversion of fertilizer into ammonia and reduce that volatilization loss. And we have nitrification inhibitors that act on these specific bacteria to try and prevent movement from ammonium into that nitrate pool that can really easily be lost through denitrification or leaching. So this is one of the tools that we use to try and control our bacteria and make sure that we're helping time our supply with our crop needs. And so this is where my research is focusing, because obviously urease and nitrification inhibitors are impacting your soil biology, and it isn't the soil microorganisms that do these transformations are also there cycling your carbon and are active in other ways. And if I'm really interested in understanding if it changes this pool. And there's also some questions I think left around how does adding inhibitors, I guess, help link that timing piece better than applying split applications to your crop? Does it improve if you use inhibitors and split application timing for, say, winter wheat? Does it improve that nitrogen use efficiency? Do you get a better yield? Is there an economic return? So there's 5 trials going in either this year, hopefully this year, if not coming up into next, like 2027 field season, that will really ask these questions and look at this from both an agronomic and a microbial point of view and a greenhouse gas emission point of view across Southern Ontario, led by Adrian Correndo, and Carrie Dunfield is, of course, a part of that. And so, yeah, it'll be interesting to see what comes of that. I did have a couple questions. I have a little bit of time about humic acid. So there was some farmers that had asked me about this, or we had talked about this briefly at in July at the Laura field day about nitrogen, about using humic acid. And there is potential that humic acid will be an effective urease inhibitor. It does not work that great as an N2O or a nitrification inhibitor based on the research data, but it depends a little bit on the humic acid. So this humic acid in this study had a carboxyl group enhancement. Basically, they just produced it in a slightly different way than the typical humic acid. In the second study, it was humic acid made from pine, and we see those reductions in urease. So potentially their humic acid might work, but I haven't seen any yield data. So hopefully there's some trials that go on in the future about this. Just before we leave, I want to talk a little bit briefly about the right place and where our losses are happening. So in these upper slope positions where it's more aerobic, so dry, and you have higher pH, you get a lot more volatilization. Whereas denitrification happens when there isn't oxygen available. So they occur in these wet areas. And so, it makes me wonder, is there potential to target the application of inhibitors where I'm applying urease inhibitors at the top of the hills and nitrification inhibitors at the bottom of hills to improve our economic return on investment, kind of in the same way that we're trying to better target the use of our herbicides or our fungicides? I don't know. There's certainly potential. This brings me back to when we apply manure. It also typically can volatilize into ammonia. And actually, we're going to talk about it here. So ammonia volatilization especially happens at the surface when there's a lot of oxidation. And so we can spray on our fertilizers, but plants take up their nutrients through their roots. They do not take it up through their leaves. So it has to be washed into the soil. So when you are applying your fertilizers, you want to get it in the ground. If you're applying UAN, that means hoping that you can time your application right before a rain, so that it washes into the ground. And if you can't, you also get a lot of ammonia volatilization from UAN fertilizers applied, on your surface. So this is where a urease inhibitor can help extend that kind of rain fast timeline where, it'll stay hopefully and give the rain an opportunity to wash it down. And same with your manure. I know it's really challenging to get it into the ground, but really that is your best way to reduce your volatilization losses. So coming back to this matrix, we all want soils in that green. And when we have soils in that blue, this is where we want to use inhibitors or organic nitrogen sources or split applications so that we improve our plant competition, and time our supply more closely with when the plants actually need it. When you have that high competition but low supply, this is where, maybe your microorganisms are not as active as they could be. Maybe that means we need to do something to feed them, or maybe there's problems with your aeration in your soil or compaction or drainage, and again, similar to the last soil, that split application, anything that improves plant uptake and plant competition over your microbes will help in this situation. And finally, when you just have a lagging soils, that's where you're trying to build your organic matter and really increase the diversity of your microorganisms with your plant, because you're trying to revive that nitrogen cycle. So yeah, today we just talked, about how our microorganisms really drive those nitrogen transformations. And we use these 4R management principles to try and close the valves in our soil. And the choices that we make, you might be using specific inhibitors if you know that you have pretty low-lying soils that hold a lot of water, and you might have a lot more N2O emissions. Whereas if you have more upland soils that are pretty well aerated, maybe you have a lot of ammonium losses, and so you're going to use a different inhibitor, or think about how you're applying your fertilizer a little differently. And again, soil health is equivalent to your soil microbial aliveness and that supply of nitrogen and sulfur and all of those other nutrients and carbon cycling that are really beneficial to your plants coming through your soils. So that's all I have for today. I hope this was